Welcome to the Nimble American Rescue Plan webinar series. This webinar series highlights the projects funded through the American Rescue Plan. The goal of these projects is to help the nation prevent, prepare for, and respond to coronavirus public health threats. In this webinar, we will hear from Dr. Manjari Lal of the PATH Center for Vaccine Innovation and Access on the project development of a thermotolerant multi-dose egg-produced vector-based coronavirus vaccine, which provides better vaccine availability by reducing storage and transportation costs. You can find additional webinars in this series on our website, www.nimble.org. And now we will turn it over to Manjari.
Thank you. Hello, everyone. Under the America Rescue Plan grant, the PATH project team tried to address the challenge with current marketed corona vaccines, which rely on complex cold chain infrastructure for delivery. These vaccines require continuous sterile refrigeration at cold or ultra-cold temperatures from the manufacturing site to syringe to maintain the vaccine stability and efficacy. These specific storage and transportation requirements makes vaccine sharing difficult between different public health facilities and even neighboring states, thus leading to vaccine wastage. Not only is the cold chain maintenance expensive and complex, but it also limits equitable distribution to underserved populations and hard-to-reach communities. We tried to address these challenges by developing a thermotolerant multi-dose vaccine, which is stable at ambient temperature conditions, not requiring continuous refrigeration. The vaccine candidate, which PATH selected for this assessment, was a vector-based COVID-19 vaccine candidate, also referred to as NDV-hexapro, to demonstrate feasibility in developing a thermostable vaccine. This specific vaccine candidate was developed by Mount Sinai and University of Texas at Austin and manufactured by Institute of Butantan in Brazil. This vaccine candidate has demonstrated efficacy in preclinical studies and several clinical trials showing acceptable safety and potent immunogenicity. PATH took this vaccine candidate and developed several multi-dose thermotolerant product formats in liquid and dry forms. These formulations were then tested for their ability to withstand elevated temperature exposure during storage over a 6-month testing period. This slide highlights the key project outcomes. PATH successfully developed thermotolerant COVID-19 vaccine in 3 different product formats. The liquid formulation was stable at 2 to 8 Celsius, which is refrigeration conditions, and ambient temperatures of 25 degrees Celsius for up to 6 months. This was the longest time point tested under this project period. The dry format maintained the vaccine content at 3 different storage temperature conditions for up to 6 months. The sublingual tablet format, which is a tablet presentation for under-the-tongue administration, also maintained its antigen content after being stored at 40 degrees for 4 weeks in this format. Multiple presentation formats of this vaccine in liquid, dry, and tablet have improved temperature stability at ambient and elevated temperatures, and they offer several potential benefits. Some of these are captured below. There is reduced cost of cold chain management in both equipment and maintenance. There is increased ease of use, especially for in the tablet format. There is reduced risk of loss of protective efficacy, which is associated with cold chain systems. This product format also allows for delivery of vaccines into underserved communities, especially in the low resource and remote areas. It also allows for increased ease of sharing between communities. And finally, the thermotolerant aspect of the vaccine provides an option of stockpiling for future needs, potentially reducing shortages. Reductions in cost with these thermotolerant formulations could potentially translate into a lower cost of the vaccine, further increasing equity in access. Thank you. Happy to take any questions.
Thank you, Manjari. Before we close, we do have a few questions. One of the things that came up from your work on this project was that 2-PE may not be a good preservative for the vector-based vaccine. Are you hoping to do further research in this?
Yes. So for a multi-dose vaccine presentation in a liquid format, it is required that a preservative be included to maintain the vaccine efficacy. And our results showed that this particular preservative was detrimental, compromising the stability of the vaccine. So in the current scope of work, we will not be testing additional preservatives, but yes, I think we would like to test other preservatives such as thimerosal or phenol, which are commonly used preservatives in other vaccine products. We would like to test compatibility of those with this vaccine candidate in the near future.
Excellent. Clearly, you had some great success with this project. What are your next steps in moving this work forward and sharing it with the scientific community?
We are drafting a publication, drafting a manuscript. There's a lot of work which went in generating this data. We would like to share it with the research community. And also the needle-free sublingual format of the vaccine is very exciting. Hopefully we can advance that product presentation further. So yeah, so we will be soon submitting the manuscript for publication.
Great. And last question, what would you say is your most significant takeaway or lesson learned from the project?
Significant takeaway, I think we should strive for making the vaccines thermostable. I think thermostability is a very critical attribute the vaccine manufacturers should incorporate or start thinking about it early on, because that can have huge impact both in terms of improving vaccine access and also cost savings. So I think I would say that is the key takeaway.
Excellent. Well, thank you, Manjari, for your time, and thank you for watching our American Rescue Plan webinar series. To find additional webinars, visit www.nimble.org.
Thank you.